Hi, let me just briefly give an overview about the talk. So I'm going to briefly set the stage for what we're talking about, because it's a little bit different, but it's also very similar. Melissa actually gave a great setup for a lot of the concepts we're talking about, and then I'm going to take you through some of the code. So just first off, again, we're not talking about disease or gene phenotypes, but about the vast space of natural phenotype diversity, which is enormously important for elucidating evolutionary relationships and the evolutionary processes that have given rise to biodiversity. And the vast majority of these natural phenotypes are documented in meticulous detail, but in natural language. And as probably many here can appreciate, natural language phenotype descriptions or trait descriptions, as we would more likely say for natural phenotypes, are highly resistant to computing. And so another problem is that all these evolutionary models or algorithms and applications that I was talking about treat morphologies as if individual traits were completely independent from each other. And we know very well that that's not the case, right? The elements in an anatomy and an organism are far from independent on each other and related to each other in all kinds of ways. So the RPhenoscape project tackles some of these challenges. It started back in 2007 with a mission to make morphology computable, discoverable, and linked to genetic data. I'm not going to talk today about genetic data at all, but really since the last few years, we've focused most of this work, and this tool that I'm presenting is so far chiefly aimed towards addressing the morphology side and making access and adoption of the morphology data more easier. So just very briefly, how do we achieve this? We take evolutionary phenotype data and model organism data and subject them through manual creation to the same standardization process using ontologies. This is a lot what Melissa already talked about, so I'm not going to spend much time here. The RPhenoscape knowledge base that's in the center here essentially follows the following architecture. Again, ignore the left-hand side about the model organisms. I'm not going to talk about that. And the important part here really to remember maybe is just that there's an awful lot going on under the hood. That's the box in the below. And I'm really going to focus the rest of this talk on this little square there in the upper right-hand corner, which is the R tool that connects to the RPhenoscape KB web API. So that's called RPhenoscape. RPhenoscape is an R package for API access to the RPhenoscape knowledge base, and particularly provides convenient access to the following things. Number one, convenient access to the evolutionary character data with computable semantics, i.e. all the ontology links. And it also provides access to the machine reasoning capabilities that the RPhenoscape KB either provides itself or actually enables to the data that it has. Specifically, these are semantics-based data filtering, trait dependency, evidence-based mutual exclusivity of traits, and synthetic supermatrix synthesis, and semantic similarity matrix. I won't really take you through much code that is about the first. That's existed for a longer time. That's not really very new, and so I'll focus on the remaining four. And so you might ask why an R package that may not be the most popular ecosystem for a lot of people working in genomics. However, it's widely popular and a very rich ecosystem for people working in phylogenetics and comparative methods. And that's really our target audience here. So getting started for an R package, this follows the usual step, except for now you have to install this from GitHub and not from CRAN, the comprehensive R archive network. And then you can get an overview, for example, about the data that's in there. And let me just clarify a little bit of the terminology here, because again, it's maybe a little bit unfamiliar for people in the genomics area. again, trait data here, trait of phenotypes. I'm going to use the two terms pretty much interchangeably. Are observable characteristics of an organism that are the product of its genome and developing and interacting with its environment, right? So this is probably not too unfamiliar. Character, and so I alluded to earlier, right? For natural phenotype data, these data are typically published as character state matrices, and in such matrices, characters are the columns, taxa are the rows, and curators link the states, which are essentially the cells to one or more ontology-defined phenotypes, okay? And so let's go into some code here. So starting character states, as I just said, right, they're linked to ontologically defined phenotypes. And so here I start with obtaining a study, and then I can get phenotypes from the knowledge base using the semantics-based filtering, right? So there are no phenotypes directly annotated in natural text to ceratobranchial 5-tooth, right, comma shape or something, right, that gets translated into ontology terms and then uses the index of the ontology terms to pull out the respective phenotypes. So that returns a data frame, which is one of the most common data structures in R, and we can look at some of the labels here that are human readable, right? I'm not showing the IRAs, they're not really human intelligible, but more importantly here, what I want to get to is that every one of these phenotypes, right, here I'm showing just one, I can turn into an object in that has all the links to ontology terms, right? So here's the character state that it's linked to. It's the bicuspid state, the state labeled bicuspid for the character ceratobranchial 5-tooth shape, right, from the study. And this is linked to the NED term from Uberon and the quality term from Pato that formalize the semantics of the phenotype. So just to keep in mind, the links between character states and phenotypes is an end-to-end relationship, really. And we can show, and this is one of the key features, right, about making the character state data interoperable across all the data matrices rather than being siloed in one data matrix, right? So we can, for example, for this one, pull together all the states and then can observe that there's now other studies here being pulled in as well, rather than the one I started with, right? That's one of the two ends that I was referring to. And also that one of the states here has links to two phenotypes, right? Because it's really combines in its description, in its natural language description, two actual qualities. And so these terms that define the phenotypes link to computable domain knowledge, and we can then leverage this domain knowledge formalized in anatomy ontologies using these relationships, right? And just sort of an example here quickly, if I use I have access through the RPhenoscape package, right, to all these relationships here, for example, for the term femur, what it is a part of, what it develops from, what it is a subclass of, and so on and so forth. And I can then use our knowledge of ontology, of anatomy formalized in the Uberon ontology to make inferences about dependence, right? In this case, presence-absence dependency. So I can here obtain a matrix that tells me if the presence of one NED or an atomic element implies the presence of another, right? So we can, for example, see that the presence of the femur implies the presence of the hind limb bud, right? Because the femur develops from a type of hind limb bud, right? And also implies the presence of a long bone, right? The matrix here is not symmetric. And so I think I need to move on in the interest of time. And so to carry on, this dependency can then also be used to infer traits based on presence-absence dependency, right? And so there's a number of things that, there's not only the presence-absence dependencies that allows us to infer, but also owl semantics, right? So in the phenotype expression, for example, the owl semantics, if I have a quality and in here's in some entity, right, owl semantics tell us that there is actually some instance of the entity exists, right? So I can infer the presence. And so we've taken this to a large scale analysis, and just to illustrate this a little bit here, I have access to this within the RPhenoscape API, right? It's called ontotrace. I can obtain ontotrace data or this inference presence-absence trait matrices for a taxon, and say here, for example, for dorsal fin across all the teleos, right? And that returns almost 3,400 taxa, whereas originally annotated presence-absence for dorsal fin only covered 370, right? So it almost increased my coverage of taxa by a factor of 10, right? And so just sort of as an aside, most of these inferences are not missing values, right? Most of these inferences are actual presence or absences. I can also use the data instead of the actual anatomical knowledge to infer or to obtain evidence for whether two phenotypes are mutually exclusive with each other, right? So there's some intuition that an organism, there's not, that an organism can simultaneously exhibit any pair of phenotypes, right? And so we can use the data to actually look at this. And so for an example, right, if I collect a number of phenotypes here about the ceratobranchial 5-tooth, I can then obtain a data structure that collects evidence from strong compatibility or strong evidence for compatibility to all the way to strong evidence for exclusivity. And so if we look at some of these, right, then we see that the absence of the tooth is exclusive with a number of other attributes or qualities of it, right? So that would be expected. But there are also some surprises in the data that can then be used to maybe look at curation errors, right? And so what I finally want to get to is that ontologies, as we've heard earlier from Lisa, also enable computational metrics of semantic similarity, right? And so just for example here, how this is accessible computationally within the RPhenoscape package, if we just look at a number of anatomy terms that will probably make a sense to most people here for illustration, using pectoral fin, pelvic fin, and forelimb and hind limb, and then dorsal fin and caudal fin, right? Here's how I can easily compute this, and then I can, instead of staring at the matrix, or I can also visualize this as a cluster dendrogram, noticing here that the dorsal fin and the caudal fin, which are unpaired fins, seem to be semantically more different from the other terms here. And so I can, let me see. So how does this work under the hood? It actually uses a very computationally efficient data structure that's that we call a subsumer matrix that essentially creates a node embedding, if you will, using the subsumers for a given set of terms that you want to compute the semantic similarity across. And so how does this look? Just as an example, right? So this is just a long list of subsumers you have as the rows, and then there's a one if the row term subsumes the column term and zero otherwise, right? And so why is this useful? It provides a really efficient matrix. It allows for really efficient matrix algebra to compare, to compute a whole number of semantic similarity matrices very quickly and without much effort using the facilities built into R already. And so we can also use this to compute profile similarity, right? Profile similarity is semantic similarity between groups rather than pairwise between single terms or nodes, right? And so, for example, here in R, we do this creating a factor. And so if we, for example, bin all the fins into one group and all the limbs into one group, and then also bin the pelvic fin, pectoral fin, forelimb, and hind limb into the paired appendage group and the other two into the unpaired, we can create an intersection and compute the profile similarity and find that paired fin, for example, are more similar to paired limbs than they are to the unpaired fins, right? Because they're homologous with each other, and semantic similarity actually bears it out. And so there's some other methods here. In the interest of time, I'm going to skip over this, but there are various ways for profile similarity to reduce the pairwise scores into a single score, right? They're all implemented here. And so what I want to get to here is that semantic similarity is also computable for phenotype class expressions, right? I showed this just for illustrations for straight up anatomy ontology terms. And so we can, for example, do this for all the phenotypes of the basically hyoid bone across the entire PhenoScape knowledge base, and see that we end up with different clusters. For example, you see here the shapes, here the sizes and lengths, and then here is some basic some surfaces, right? So you can really visualize the similarities between the different phenotypes being more closely or being more distantly related. We can then also use the phenotype semantic similarity to compute something like semantic information content across characters or even across studies and compare them from a semantic standpoint, right? So here I'm doing this for studies, right? All the studies that have phenotypes about the basic hyoid bone, and I've used just those to compare semantic similarity profiles for each study. You know, some studies align with each other much more closely than others. And so that was a walkthrough through the major parts, major computational capabilities of the RPhenoscape package. And I want to just sort of at the tail end of my talk, go through a few thoughts as to how it ended up relatively successful as it did. So number one is we started with driving use cases extracted from full phrase user stories that describe the personas, why they want to do something, how they want to do something, you know, that gives context. So that was very helpful and at hindsight. Also, a strong focus on usability. So that requires understanding your audience, which is why user stories are really important to start with. And you know, that entails, once you understand your user audience, right, is how is the computational environment, the ecosystem that they normally work on, and make it as easy as possible for them, as seamless and barrierless and frictionless, I guess is the term I'm reaching for, as possible for them, right? One thing here that I really want to mention that it gets often underestimated is what proved to be very powerful is an efficient and highly parameterized term finder, right? It sounds very trivial and like really a sort of plumbing, but found very important. And so that extends to, you know, more complicated things like, for example, class expressions, where you can input a natural language description and then obtain it fully turned into IRIs. So some control over the database API also proved useful, and I think I'm about at the end of my time here, and finally, actually, users as part of the project, right? Really important, rather than trying to develop for people for hypothetical users. Current limitations, just briefly, it's not yet on CRAN, so you need to install from GitHub. The PhenoScape data, the PhenoScape QB content is concentrated vertebrates, and fin limb characters mostly, have some others too, but that's where most of the data is. And the matrix synthesis is currently limited to presence absence. Yeah, so in summary, RPhenoscape is this bridge between computational morphology data and computational and the semantics capabilities of the Phenoscape KB and the whole ecosystem of comparative phylogenetics. People, developers, researchers working in R. It translates between R, right, and the KB API, which maybe sounds trivial, but in reality, when you try it yourself, it's actually far from it. And yeah, it's sort of an enabling tool for research approaches, right? So with that, I'd like to acknowledge the funding from NSF of the early stages of the whole Phenoscape project was incubated and for many years hosted at the National Evolutionary Synthesis Center, which does no longer exist, and finally to other Phenoscape contributors. Thank you.